I'm Rayan Chattarjee from Stanford, and today I'm going to talk about determining the hair bundle properties from physiological measurements in outer hair cells. So I'll briefly introduce the hair bundles first. So this schematic shows how the sound wave vibrations it transmits from the outer ear via the middle ear and then to the inner ear, and where the sound sensing organelles are located, and they transduce these vibrational forces into nerve signals of the brain. So now this is a zoomed in view of the organ of Corti, the sensory apparatus of the inner ear, and it highlights the hair bundles. The hair bundles are the mechanosensors within the organ of Corti, within the inner ear, and they're located on the top of the hair cells, which are actually the mechanoreceptors, and they convert the sound-induced vibrational forces into receptor currents by a process which is called mechanoelectrical transduction. So the signals from the inner, there are two types of hair cell I'd like to highlight here, the inner hair cell, the outer hair cells. So the signals from the inner hair cells, they are sent to the auditory nerves, which are finally transmitted to the brains. The outer hair cells, the electrical signals from them, they work to amplify the signals which is sent to the inner hair cells. So today I'm going to talk about the hair bundles of the outer hair cells. So this is a zoomed in view of a single outer hair cell, highlighting the hair bundle at the top. So if I zoom it further, I can see the components are seen. So a hair bundle consists of rod-like stereocilia, which are pivoting on the hair cell apex, the gating springs, which connect its stereocilium to its nearest shorter neighbor, the ion channels, which lie at the apices of its shorter stereocilium, and their opening or closing is regulated by the tension in these gating springs. So the model that we are developing for the outer hair cell hair bundle, the height of the longest stereocilium is 4.1 micrometer, and the stereociliary width is 200 nanometer. So the basic, in simple words, the function of the hair bundle is mechanoelectrical transduction, or shortly called MET. So what is in brief, the sound-induced forces, they deflect the stereocilia, which extends the gating springs, which opens up the ion channels, and as a result, the potassium ions from the surroundings, they flow into the hair cell, thus setting up the receptor currents. So the hair bundle we'll show the data for most of this talk. It's oscillating at the oscillates at the frequency of sound around 4 kilohertz, which is faster than most cells. And the big question is, how do the components of the hair bundle contribute to its function? So before telling our strategy, I would again like to highlight some unique features of hair bundle and some challenges in hair bundle research. So hair bundles are highly sensitive. The deflections in response to the quietest sounds that we can hear, the hair bundle moves a distance which is less than the cell membrane thickness. Also, the maximum deflections are of the order of the stereociliary diameter. Secondly, the response of the hair bundle is extremely fast because the mechanoelectrical transduction, it takes less than a millisecond. And some of the challenges are, a hair bundle is a 3D, if one talks on the terms of modeling, a hair bundle is a 3D complex system, and over 100 coupled nonlinear equations of motion are required to describe the motion of an outer hair cell hair bundle. And secondly, the experimental procedures, they're very complex, and there are uncertainties in measurements of both the morphology as well as the material properties of the hair bundle. And also the results that we get from experience, they're very sensitive to age of the sample, the species of the sample, and various other factors. So our current strategy is to build a computational model of the outer hair cell hair bundle which takes into account the bundle morphology. So thus, using this model, we will determine we are determining the minimal parameter set within which the model response matches that of the experiments. And secondly is to investigate the effect of the uncertainties in measurements on the estimated values of the parameters. So now, I'm going to briefly describe some of the properties of the hair bundle model. So the model that I'm going to discuss today is a simple one-dimensional model, and it consists of 3 stereocilium, 2 gating springs. The stereocilium are named 1, 2, and 3 in decreasing order of heights. So the properties, some of the properties are directly measured. For the heights of each stereocilium, the stereociliary widths, the distance between pivots, and the deflections owing to external forces, and some of the properties which are not directly measurable from experiments, but we can calculate them using our model. So for the pivots, we assign a stiffness, a damping coefficient, and an unloaded angle. And for the gating springs, there is a stiffness and an unloaded length too. And in the remaining of the slides, I will briefly go into a bit of the details of how do we calculate the stiffness of the pivots as well as the gating springs. So to determine the pivot and the gating spring stiffness using the experimental data, the data that we use are mostly from the paper by Tobin in 2019. So it is basically generated for a hair bundle of the rat hair bundle using the procedure is to mechanically stimulate a hair bundle using a calibrated fluid jet, and then to record the deflections produced in response to each force. And here is the load versus force versus displacement curve for the hair bundle with for the full hair bundle, that is the stereocilia plus the gating springs. So we use this as one of our input data set, and the second data that we use where the experimental procedure is the same, but the gating springs are disrupted using a chemical called BAPTA. So here the load deflections are recorded for the hair bundle without the gating springs. And we use these two data to determine the pivot stiffness and the gating spring stiffness in our model. So next, I'll now describe how the agreement between our model and the experimental data. So first, I will discuss the model without the gating springs. So in absence of the gating springs, the force balance is between the external force and the pivots. So as I highlight here, the generalized formula of the pivot force, it depends on many terms: the pivot stiffness, the angles at rest, and the lengths of each, the heights of each stereocilium. And here is the plot which shows the agreement between the experimental data and our model. So the red dots here, they correspond to the experimental data from Tobin's paper, and the continuous red line, it indicates the fit from our model. So so to do so, we adjust the model parameters to match the experimental data. And for this particular case of the hair bundle without the gating springs, the pivots are the only structures that provide elastic resistance. And using this physical information, we can calculate the stiffness of stereociliary pivots for our model. We calculate the gating spring stiffness. So here is the force balance equation. So as I emphasize here, that with the gating springs, the external force is balanced by elastic force given by the pivots as well as the gating springs, and both are dependent on the length of the stereocilia as well as the stiffnesses and other parameters. So here is the match of the experimental data with our model. So the black dots, it indicates the experiments from Tobin, and the continuous black line, it indicates our model fits. And we find that in both cases, with versus without the gating springs, our model, we are able to get a good match between the experimental data and the model. So one thing to highlight here is that in absence of the gating springs, which is indicated by the red curve, the stereocilia deflections are less. With the gating springs, the stereocilia deflections are more because the gating springs offer additional resistance to the force. So now I will discuss in brief the effect of the uncertainties in measurement on the measured properties. So I again rewrite the force balance of the stereocilia and the gating springs in response to an external force. And so here I emphasize that both the both the forces are dependent on the length of the stereocilia. So in order to test how much the variation due to uncertainties in morphology, it affects the estimation of the stiffnesses, we vary the height of the stereocilium 1 within one standard deviation or about the mean, and we get this range from the experimental data. And the ratios of the other two stereocilium, they are scaled by a factor which is set by electron microscopy. So this plot is it shows the variation in the gating spring stiffness as a function of the stereociliary height, stereocilium 1 height. This red dot, it indicates the value that we calculate for the mean value of the stereocilium height. So what we observe here is that there is around 4,000% variation in the estimated value of the gating spring stiffness that we observe about one standard deviation of the mean height, which indicates that the uncertainty in the gating spring stiffness measurement that corresponds to this morphological uncertainty is around 4,000%, is greater than 4,000% in fact. And for the pivot stiffness, this is shown in the second plot. We plot the pivot stiffness as a function of the stereociliary height, and here also we see that we get around 175% variation within the range of within the standard deviation, which indicates that the estimation of material properties of the hair bundle are very sensitive to uncertainties in the morphological measurements, and we need better estimates, better error bounds on the morphological data. So our next step is to extend our 1D model, one-dimensional model, to a three-dimensional model. As the right-hand plot shows, this is a electron microscopy picture of a full three-dimensional hair bundle which consists of multiple columns which are interconnected. What we are planning to do is once we establish the parameters for our one-dimensional model, we will extend it to 3 dimensions and calculate its response in response to external forces, and we'll compare it with the one-dimensional model.